The year is 2050, and there is a war raging around the globe, leaving millions of casualties in its wake. This war is not occurring between people, but within them. I am referring to the battle between medicine and antibiotic-resistant bacteria. And unfortunately, medicine is losing. Antibiotics are no longer effective at treating bacterial infections. It is predicted that by the year 2050, there will be 10 million deaths worldwide each year due to antibiotic-resistant bacterial infections. We need alternative treatments if we are to avoid yet another global pandemic. But there is hope using viruses. Now I know what you're thinking. Viruses, like COVID-19? Is she crazy? No, I am talking about bacterial viruses, known as bacteriophages, or phages for short. These viruses infect and kill only bacterial cells, and different phages are specific to a single species of bacteria. This means we can select phages for therapy with a specific arsenal designed to target and kill only the pathogenic bacteria causing disease, and leave beneficial bacteria unharmed. Now, how do we choose phages to treat a given bacterial infection? Well, that's where I come in. My research studies the interaction between bacteria and phages. You can think of this interaction like a lock and key. There are many locks on the surface of a bacterial cell, and a phage may have one of several keys. However, each key will fit only one specific lock. So it is this interaction that dictates which bacteria a phage can infect and kill. I can identify the receptor, the lock on the bacterial surface that a phage is binding to, and ultimately use that information to create mixtures of phages targeting multiple different receptors. In this way, I target the bacteria from all angles and limit the evolution of bacterial resistance to a phage treatment. I also investigate the phage proteins used to bind the bacterial cell. These phage receptor binding proteins, their keys, are present on the tails of the phage particle. By identifying these proteins, I have a toolbox at my disposal to create phages targeting a wider range of bacterial hosts. I can do this by exchanging the genetic material the phage needs to make those receptor binding proteins, or in other words, exchange what key that phage has, and engineer a phage better equipped to target and kill deadly bacterial infections. So, in the war against the coming antibiotic resistance crisis, let's unlock the solution using phages.